Hi folks, this is Mr. Ferrari, and this is just going to be one of two quick videos to quickly review just some of the topics that I said I was going to review for you, and so you've got something that you can work with. I would also encourage you to find other resources outside of myself. Sometimes it's better to find other people that are discussing the same things because they might explain it in a little bit of a different way that might help you out a little bit. So don't mind these; those are just testing pens. So the first thing we're going to look at is this idea of mitosis versus meiosis. And the major thing we want to talk about and we want to remember is that mitosis is merely for asexual reproduction, or growth and development. So maybe something like a bacteria will do some sort of asexual reproduction, or maybe we're talking about, hey, we've got a bunch of skin cells here, they're getting a little large, so let's do some mitosis so we're not getting too big. On the contrary, we got meiosis, which is for sexual reproduction. Sexual reproduction. And that's it. Meiosis is necessary for sexual reproduction. Now, of course, meiosis isn't the act of sexual reproduction, but more so what's needed for sexual reproduction. The big thing is that this will lead to large genetic variation, and that is key in this process. Large genetic variation. So just to quickly review mitosis here. In mitosis, if we had a cell like this, and this is going to be a very generic drawing, very generic, but it might have some DNA, and its DNA might have already been replicated. So here's its DNA. It would begin the process and enter prophase, which actually we can call this. I see condensed chromosomes. There's no nuclear envelope. We can throw some centrosomes here. Why not? Make it look a little bit more like it. So we can call it prophase. And then we see in metaphase that these guys will line up in the middle. Remember that meta is middle. So that the centrosomes here, we'll pull them apart. Well, not yet. That's next. They'll connect at the centromeres of the chromosomes. The centrosomes will connect to the centromeres of the chromosomes with the spindle fibers. So that during anaphase, these will all get pulled apart. And again, I draw them with an inverse direction now because this is supposed to be showing some sort of pull movement occurring. So there's some sort of pulling occurring so that in anaphase we can see that they're getting pulled apart. And in telophase, this is where we see that cleavage furrow come in. They're all kind of over here doing their thing. Perhaps the nuclear envelope is starting to come back on each side. Right, and like I said, this is starting to cut. There's that cleavage furrow, that's telophase. So in cytokinesis, these will become two separate cells, but they're genetically identical to this guy before these were reproduced. Because there's actually going to be another one that's right here. It's going to maybe look like this. It looks a little bit like that. We can maybe call this interphase or something of the sort. And that's quick mitosis. We know that. That's good. Now meiosis, I'm just going to do a different piece of paper here. Meiosis, I've talked about is this idea of like it's mitosis but twice. And that's not fully inaccurate. When I say it's mitosis but twice, I mean that it's the same thing in that it's PMAT. There's prophase, metaphase, anaphase, and telophase, and it does that twice. Right? We've talked about this idea of prophase 1, metaphase 1, anaphase 1, telophase 1, etc. for meiosis 2 then, so it's prophase, metaphase, anaphase, telophase, all 2. The difference is what we kind of glanced over here in mitosis is this idea of homologous chromosomes. And remember that a homologous chromosomes are the pair of the same chromosome. And when I say that, I don't mean that it's exactly the same thing. It could be a little bit different. And this plays into the idea of alleles and genotypes. But when I talk about pairs of the same chromosomes, I'm talking about those karyotypes that we talked about in class. So if I had a rough little draft of a karyotype, this might be a little something of a, some organism that has, let's call it 9 chromosome pairs, or 18 total chromosomes here. There are 9 pairs. So we get 18 total. And I did include something that looks a little something like an X chromosome and a Y chromosome here on purpose. So we can see that as well, because they are not homologous, but we can kind of think of them the same way. So these are our homologous chromosomes. And again, this is just a rough sketching of an organism's karyotype that might have 9 pairs or 18 total chromosomes. We know that in humans, it's actually 23 pairs, and so we have 46 total chromosomes. And so I want to stress this idea of haploid and diploid. These are haploid numbers. These are diploid numbers. When I say haploid or diploid, I'm talking about the number of chromosomes. So in this karyotype here that we're looking at here, this is the karyotype of a diploid cell. This is diploid because there is not one, but 2 versions of each chromosome. There is a 1A and a 1B. Now, if I do something like this, something a little along the lines like that, then I would call that a haploid karyotype. So this is a diploid karyotype, and this is a haploid karyotype, right? There are 2 versions of each chromosome here. There's only 1 version here, haploid, like half. And we might see this referred to as 2N and this as N. N is talking about our haploid number. So keeping that in mind, like I said, one of the things that we kind of glossed over in mitosis we just said the X's get split up. And that's accurate. But we didn't talk about the fact that there's actually an X for each one of these, even if it's the same chromosome. So now we're actually going to have, and I'm going to go back to this 4 chromosome, or let's call it 2 chromosome. There's 2 chromosomes here. Let's call this one A, 1B. I'm going to go back to this 2 chromosome idea here. This haploid would be 2 and the diploid would be 4. So I've got a cell here. I've got a cell in the nucleus. I've drawn 4 total chromosomes, and this is a diploid right now. This is an organism that has 2 versions of chromosome 1 and 2 versions of chromosome 2. That's it. Fairly simple, but that's it. So that if I were to draw this as replicated, there's a homologous chromosome pair, and there's a homologous chromosome pair. And these are technically homologous as well. There's still 2 pairs of the same chromosome, but now we've replicated them. Now we've created replicated or duplicated homologous chromosome pairs. So we can call this interphase here, and let's call this prophase 1. This is now prophase 1. And so what we'll see here is we'll see our nuclear envelopes having dissolved a little bit, it's starting to fall apart. Centrosomes are showing up, sure, that's fine. And so that in metaphase 1, this idea of meta is middle is still viable. But now we're going to line up a little bit differently. Here are my centrosomes. Notice how instead of lining them up completely vertically like we did in mitosis, I've now matched them kind of next to each other. These are the homologous pairs again. This would be chromosome 1A, this would be chromosome 1B, this would be 2A, and this would be 2B. So that now during metaphase, as those spindles start to attach to the centromeres, we see in anaphase 1, this is anaphase 1, the actual X's are getting pulled away from each other. They're not getting broken yet. They're not getting broken like we've seen before, especially with mitosis. We just see the X's split from each other. This is the point in time where a diploid cell becomes haploid. If this was a diploid of 4, this is now a haploid of 2. There is 1, 2, 3, 4 chromosomes, so we can call this a diploid 4. But now if we're going to start separating here, this is where we can start to see our diploid become haploid. Now, granted, this will actually happen after telophase and cytokinesis, but this is where this idea is coming from, now that we've split 1A from 1B and 2A from 2B, so that in telophase 1, and that's kind of a rough drawing. Ooh, that was something. We've got one chromosome there and another one there. There's our cleavage furrow. Our nucleus is maybe coming back just a little bit, just so it can get broken up again. And there's our telophase 1. So that now, at this point, now we're going to get 2 cells. Now we're going to get 2 cells, and let me slide this up a little bit. I got some stuff here that I need to move. Pull this back down. Wonderful. Now I've got 2 cells here that both have 1/2 of the original contents. Oh, I forgot to draw chromosome 2 there. Okay, now we've got 2 cells that have 1/2 of the original cell's contents. Here's chromosome 1A, and this is now chromosome 1B. This is 2A, and this is 2B. Okay? And now this is where this process of kind of becoming mitosis kind of comes back. Meiosis 2 is very similar to meiosis in the sense where, we can call this prophase 1 here. That's fine. We'll just skip going back into interphase because it really doesn't happen. I'm sorry, this is also prophase 2. I misspoke. This is prophase 2 here for both of these cells. So that in metaphase 2, now we'll see the X's just line up vertically like we know from before. Centromeres are here, they're doing their thing, all good, nice and good. Nothing's going wrong here, no non-destructions today. This is our metaphase 2. This is our metaphase 2 here. Hopefully you can see that. Maybe I'll pull this up a little bit more just in case. Okay, a little bit more. Sorry, there's a bunch of keyboards on this. There we go, much better. Now we're in metaphase 2, and so we'll just follow the same process that we know from before. Like I said, this is where it becomes very mitosis-esque in the sense where everything that we're going to see from now on is actually what we just saw here. So our X's will get pulled apart like we know from before. There's my centrosomes. They're going to be pulling apart these X's, and again, I'm going to draw them inverse this way to show that there's a little bit of inertia because they're getting pulled. Oh, drew them backwards. This is supposed to be 1. Okay, they're getting pulled away to the opposite sides like normal, like that. And so that in telophase 2, now we've got 2 cells that are undergoing telophase. There's our cleavage furrow in each one of them, right there. Nucleus is starting to come back in each. This is telophase 2. So the end products of meiosis are now 4 total gametes. And again, I say the word gametes because that's the fancy word for a sex cell. That's the product of meiosis. Each of which that have half of the original DNA of the one parent cell. They have half of the DNA that we saw here. This is chromosome. This is one version of 1A. I'm sorry. This is 1A. This is 1B. This is 2A. This is 2B. These are 1A. These are 2A. These are 1B. These are 2B. These are a result of these guys, right? Of these guys that became X's from the original. That's why we're going to get 4, is because now there's actually 1, 2, 3, 4 chromatids that all go along with chromosome 1. But 2 of them are now from chromosome 1A and 2 from chromosome 1B. And the same thing here with chromosome 2. I've got 2 from 2A and 2 from 2B. So there's 1 from 1A, the second from A. Here's the second from 1B and the second or first from 1B and the second from 1B. And then I've got, 1 2A, another 2A, 1 2B, and another 2B. And so these are our gametes. These are our gametes. This is the product of meiosis right there. We just created sex cells. Again, the process of this is that now in human cells, each one of these is 23. And so that should a sperm cell find its way to an egg cell that also has 23, when they fertilize, then we'll get our diploid number of 46. And so we'll have a healthy normal of chromosomes. And it's this separation, especially the fact that this is generally random, that leads to a lot of genetic variation that we see and that we'll see here in the next video. So hopefully that helped. Again, I highly encourage you, go find another resource. Don't just use my stuff. Find another resource. I'm sure it will help you to hear it in a different voice. That being said, thanks for watching this one. I hope this helped. Good luck this week. Good luck on Thursday.